See, we biologists perform experiments, and as a result of that, we are able to collect data, or we may be collecting data from the population regarding certain aspects, and that data has to be analyzed. Statistical analysis uh, that is required to reach on a specific inference. So we apply a number of statistical tests like student t-test or analysis of variance, that is ANOVA, or g-test or chi-square test or Mann-Whitney U-test. So these are different statistical analysis. But before utilizing a specific test, we have to actually establish a hypothesis, and we have to test that hypothesis, that whether the hypothesis comes true in the present case or not. So hypothesis testing begins with an assumption. We have to actually assume, or we have to consider the established theory or hypothesis, and accordingly we have to test that. So hypothesis testing begins with an assumption called a hypothesis that we make about a population parameter. And a hypothesis is a supposition made as a basis for reasoning. The hypothesis could be null hypothesis. Statistically, actually, we decide two types of situation: one null hypothesis and the other alternate hypothesis. See, null hypothesis means there is no difference between the observed and expected conditions, or between the two samples. So no variation exists. We consider with this, null hypothesis means no difference is there in the collected data and the data which has already been procured earlier. If we have to make comparison between the two, then that assumption we have to make. And if we do not consider this null hypothesis, then the other one will be alternate hypothesis. It will be just opposite to the null hypothesis. You can understand this aspect by going through this example. See, length of right and left wing of an insect species is measured. We want to test whether there is difference in the size of the wing of the two sides. Please try to understand what is given here, that we have a specific insect species. It is winged insect. Right and left wings are present in the insect. We want to see that whether the wings of both sides are exactly similar in their length, in their size. So we shall be measuring the length of the right wing, maybe of 10 or 20 insects, and exactly the same number of wings of the other side will also be measured. And then we can apply, t-test to see that whether the size of wing of both sides differ significantly or not. So we establish actually a hypothesis, and in the beginning we will be considering that the wing size of right and left do not vary from each other. So we hypothesize that there is no difference in the right and left wing size, and this becomes our null hypothesis. The alternate hypothesis will be just opposite to it. That is, there is difference between right and left wing. Now this particular slide is of much use. You can understand the different steps which are involved in hypothesis testing. So procedure for hypothesis testing is given here. First, you decide the null hypothesis. That is, if two samples are there, or if two variables are there, then you will be considering that these two samples have come from the same population, or significant difference does not exist between the mean values of the two samples. So first you have to actually decide the null hypothesis, and then just contrast to that there will be another hypothesis called as alternate hypothesis. Null hypothesis that is being depicted by H naught. H stands for hypothesis and O stands for null. So null hypothesis. Alternate hypothesis will be just opposite to it. It means the two samples, they have a significant difference in their mean values, and this is indicated by HA. Now after deciding the hypothesis, what you will do is state a significance level. That is, you decide that at which level you are going to test this hypothesis. At 5% level, 5% means at 0.05, or at 1% level, that is 0.01. And then, so this will be the alpha value. Alpha will be at 5% or 1%. This you can also decide. In most of the cases, or I can say that in 99% of the cases, actually the significance level becomes 5%. But this is our own decision. And then decide a test statistics, whether you are going to apply student t-test or z-test or f-test or chi-square test or Mann-Whitney u-test. So this you have to decide that which statistical analysis will be most appropriate. The next step will be calculate value of test statistics. If you are doing student t-test, then t value has to be available. So whatever statistical analysis you are doing, the value of that specific test will have to be obtained. So that will be the calculated value. And then find out the p-value at a given significance level from the table. That is, for every such statistical analysis, specific tables are there, like chi-square tables are there, student t-test tables are there, f table are there, and in such table degree of freedom and values at different significance level will be given. So you have to find out the table value at a specific degree of freedom, and after that you will be comparing your calculated value with the table value. So compare the p-value, or that is table value, with the calculated value. And here is the important thing you have to consider, that if your p-value, that is table value, is more than the calculated value, then you will accept the null hypothesis. This is important thing. In most of the cases, if the p-value, which is referred as p critical or p table value, if this value is more than your calculated value, then you will accept the null hypothesis. And if this p-value is less than the calculated value, then you will reject the null hypothesis. You will be accepting the alternate hypothesis. So this aspect is quite important, because you have to make a comparison between the calculated value and the table value. So on that very basis, the null hypothesis will either be accepted or it will be rejected. Another important thing you should know, that there are two types of errors in testing the hypothesis. That is, when statistical hypothesis is tested, then there are four possibilities. The first possibility will be the hypothesis is true. Whatever hypothesis, null hypothesis you are deciding, that is true, but our test reject it. Means your analysis that is not accepting this hypothesis. So that is type 1 error. Once again I am repeating it. You have decided that the ratio of the two events will be 1:1. So that is the null hypothesis. That is the male-female ratio in the population will be 1:1. But you do not get this ratio if you collect the data and you find that this is not really happening. Then that will be type 1 error. And this may happen because of certain reasons. This may happen that your hypothesis may not get accepted. Second situation will be the hypothesis is false, means you are deciding a hypothesis which is not correct, but you are accepting, means your results help you to accept it. That is actually type 2 error, and this is actually more dangerous situation. Third situation may be that the hypothesis is true and our test also accepts it. That is, you are considering that the ratio of male female in the population will be 1:1 and your data also suggests the same thing. Then this is actually correct decision. And the fourth situation, the hypothesis is false and your test is also rejecting it. So this is also a correct decision. So these four possibilities could be seen with the help of this table. Exactly the same thing is mentioned here, that you have decided a null hypothesis and another one is alternate hypothesis. So null hypothesis is true and null hypothesis is false. That is shown here on the left side. And on the upper side, your data shows either acceptance or rejection of the hypothesis. So if you are having null hypothesis and if it is your data also shows the same thing according to the hypothesis, then it is correct decision. And in case if the null hypothesis is correct but your data doesn't actually show such kind of thing, so you are rejecting actually null hypothesis, and this is type 1 error. And your hypothesis may be false but you are accepting it. If this is the situation, then accept a false null hypothesis, so it is type 2 error. And in case the null hypothesis is false and you are also rejecting it, so it is correct conclusion or correct decision. So these four possibilities are there. Quite often we try to understand the meaning of type 1 and type 2 error. Type 1 error may happen because sometimes because of certain regions, your established hypothesis may not be accepted. So this may happen. But this situation type 2 error is that should not happen actually. So this is more serious one.